Thank you, Sanjay. Well, this is my first time at ICTS, and I'm jealous of those of you who have visited before, but I'm delighted that I've had this opportunity, and I thank Rajesh for inviting me, and I want to congratulate Spenta for his extraordinary leadership, and now Rajesh for carrying the flag forward. Having built a little bit of an institution myself, I'm completely sympathetic to all that Spenta and now Rajesh have gone through. And really, my warmest congratulations to you for the first 10 years for building a wonderful institution. Uh, um, and my best for the next 10 years. I must confess, a little bit of the motivation for my talk is to persuade Rajesh to sort of go through on the next sort of hexagonal vision of his that should include biological computation, and I hope that includes neural computation or neuroscience as one of the pillars of the biological area. All right. So brain processes underlying cognition, and the function of the brain is to create the mind, fundamentally involve information representation and transformation. And what is cognition? As defined by the Oxford English Dictionary, cognition refers to the mental action or process of acquiring knowledge and understanding through thought, experience, and the senses. And more commonly, cognition simply refers to what the mind is and does, which is mental activity underlying perception, learning, memory, decisions, action, communication. How does the brain create cognition? I think this is the deepest question in the field of neuroscience and cognitive science. For today's talk, we work on many things, but today I'll talk about vision and our work on how vision might be created in the brain through underlying circuits. So vision starts with the eyes, but the eyes are just the front end of vision. The eyes send the brain pixels of information. Actually, it sends more than pixels. It sends a filtered view of the world in a specially filtered way. But from that filtered view, there is nothing intrinsic in that view that says, here is an object, here is Rajesh, here is Spenta, here is Sanjay. The brain creates the view that one interprets as vision. And this view is created, vision is created through a hierarchy of areas. Visual cognition arises from information processing by specific cortical circuits that abstract information, throw away massive amounts of information, and then routes that information in successive stages in which more and more components of vision get extracted, if you will. We heard a little bit of this from Jennifer in sort of in passing. And out of that, the brain creates an internal model, which we call vision. It derives from the world, but it's far from a mirror of it. And in fact, I think our first speaker this morning referred to David Marr, and indeed, Marr begins his book with perhaps what is one of the most profound questions in our field. It's a simple question. What does it mean to see? And Marr goes on to outline a three-layered program that for a problem that the brain has to solve, one can think of it at a computational level, well, what defines the task that has to be done to solve the problem, the algorithm or the rules that have to be implemented, and the actual hardware implementation. I work on these two levels, algorithmic and implementation, because I'm an experimentalist, but I do believe, in agreement with Sri Kulkarni, that neuroscience, just like astronomy, is a phenomenological science. All of biology is. It's a story of history. We got here because of the history that our species has gone through. And so experiments provide the driver of information for our field. I'll show you experiments because that's what we do. I'll tell you about how we do experiments so that you understand how we know what we know. And third, so that the conclusions, the broad conclusions that I draw at the end, may have some standing in your mind. What is an appropriate level to study brain mechanisms? Surya has talked a lot about neuronal dynamics and dimensions, but implicit in what Surya says is something deeper. We can study the brain at the level of synapses and molecules that make up neurons, because neurons are really cells, just like any other cell in the body. Or we can study them at the level of individual neurons, or at the level of circuits, the connections made by neurons with each other. or at the level of large-scale systems, such as we record with functional magnetic resonance imaging. And my bias is that neuronal circuits are the correct level of organization, and single neuron activity reflects their participation in circuits. We have known the primacy of the neuron for 100 years when the Spanish anatomist Ramon y Cajal made these beautiful pictures from Golgi stains for which he was given the Nobel Prize in 1905. And Cajal himself had this extraordinary insight that this is the unit of information processing, not the molecules in the neuron, not the synapses, and not hundreds or millions of neurons, but the neuron. And the reason he thought so is that neurons make connections with other neurons in order to make circuits. We understand, I believe strongly, that circuits are the engine of the brain. And I'll tell you today about cell-specific circuits, circuits formed by the connections between excitatory, or these neurons in triangles that you can see even if you squint in Cajal's pictures, which are called excitatory or pyramidal-shaped neurons, versus the neurons in blobs, which are inhibitory neurons. There are two fundamental types of neurons in the brain. There are many kinds of these, and there are many kinds of these, but physiologists tend to be lumpers. So we tend to lump them into at least these two important functional kinds, and their interaction leads to tuned responses, response gain control, coding of information, functional dynamics, and I hope in the next 20 minutes I'll be able to give you a brief glimpse into how we decipher the circuits that do these interesting things and why they matter. I'll take you through three questions quickly. How do cortical circuits create feature selective responses? What that means will become clear in a minute. How do cortical circuits create neuronal spike codes? How do cortical circuits generate an internal model, in this case, of a task that a mouse we can train to do, in which there is working memory and sensory motor transformation? How does the mouse model this task in the dynamics in its head? And I hope you will get that you will go away with some appreciation of the nature of brain processing and the nature of questions that still our field needs to frame first and then solve. Okay, so how do cortical circuits create feature selective responses? I emphasize that vision starts with the eyes but is created in the brain, and there are critical properties of the visual cortex neurons that underlie vision. We see the way we do because there are circuits in our brain that make responses possible, and one of these responses is called orientation selectivity. It is such an important property that two neurophysiologists, David Hubel and Torsten Wiesel, at Harvard, unfortunately, were given the Nobel Prize. I say that because there is this relationship that MIT and Harvard have for a long time. They were given the Nobel Prize for discoveries they made in the late 50s and early 60s on the nature of information processing and visual processing in the primary visual cortex, or V1. They discovered the property of orientation selectivity, which is that a single neuron in the brain responds not to a spot of light or to light everywhere, but to an oriented light and dark edge. Why is that interesting? There are many, many, many reasons why they're interesting, but now the most important is from this paper by Bruno Olshausen and David Field, where they showed that if you take a natural image like this, orientation selective cells are highly efficient filters for sparse representation of natural images. The brain goes to great trouble to minimize the number of spikes it puts out. Every spike is highly energy demanding. Spikes occur by sodium rushing in down its concentration gradient and potassium rushing out, but then that sodium has to be pushed back out and potassium has to be pushed back in by transporters, and these they suck up energy. So the fewer the spikes, the better. Hence, a great problem in neuroscience is how can you convey the maximum information with the fewest spikes? I thought I'll emphasize some first principles, such as they are in neuroscience, so that you may reflect on them as ways to how can we build models of brain function. And so there have been many examples in the literature of orientation tuning. Here's one that my first student, Anna Rowe, now the director of an institute in China and a professor in Oregon, recorded by sticking an electrode in the visual cortex of a ferret. And she showed this ferret bars or gratings of light at different orientation. And here are two neurons, one that responds best to a bar of light at about 10 o'clock, 4 o'clock, and moving up and to the right. And here's another that responds to a differently oriented bar of light. A question that I asked a long time ago when I first started my lab at Yale before I came to MIT was, how do orientation selective cells and maps arise? And we did an experiment that was seen as outrageous, but has since then provided a lot of useful information. Vision occurs by the eyes projecting the visual thalamus to the visual cortex. Audition occurs by the ears or the cochlea projecting through auditory brainstem centers to the auditory thalamus to the auditory cortex. And I asked what is intrinsically visual about the visual cortex. If I can make the eyes project to the auditory thalamus and hence drive the auditory cortex with vision, would there be orientation selective cells here if these cells are so driven by the nature of the world, which is itself made up of oriented lines? That's the intuition from Olshausen and Field. And so I took newborn ferrets. Here's a painting by Leonardo da Vinci. It's called Portrait of a Lady with Ferret. It hangs in the Czartoryski Museum in Krakow. I went to Krakow just to see this painting, and it's astonishing. Why a ferret? Not only does the ferret have a wonderful visual system that resembles ours with frontally placed eyes and so forth, but a baby ferret is born at a state that is very early in development, at the equivalent stage of the first trimester of birth in humans. So you have access to very early developmental stages when axons are still connecting to their targets. So you can manipulate targets and rewire the brain. And so that's what we did. We took newborn ferrets, we removed the auditory inputs, which much later, it took us 20 years to fully understand this experiment. But in the first paper itself, we knew that something has happened to the auditory thalamus so that the early axonal pathfinding has changed, and the retinal axons are projecting to the auditory thalamus, thereby providing a pathway to the auditory cortex. We let these animals grow up, and then we asked, were there visual cells in the auditory cortex, and if so, what were their properties? And we found that indeed there were orientation selective cells in the rewired auditory cortex. And not only that, in the normal visual cortex, there is this beautiful compact solution to the problem of representing all orientations within each local region of the cortex. We see all orientations everywhere we look. And the cortex solves the problem of a retinotopic map combined with feature selectivity by creating pinwheels, where within each local region of the cortex, there are receptive fields. These neurons have receptive fields in one local region of the retina, which looks out onto space if you fix your eyes. Within that local region of retina, there are retinal ganglion cells that contribute to all orientations. And there are self-organizing feature map algorithms that show clearly that due to local excitation, long-range inhibition, you will get pinwheels. And the rewired auditory cortex that was never made to see creates orientation selective cells. These are just the color codes that simply say that a pixel here has this orientation, a pixel here has this orientation, and so forth, when we record using optical imaging, and that's a tool for imaging the brain. How might this be possible? And underlying this map is a nature of connectivity. In the normal visual cortex, these regions connect to each other, iso-orientation regions. In the normal auditory cortex, in a normal animal, isofrequency connections happen in the cortex. But in the rewired auditory cortex, the connections between neurons is reorganized. It resembles that in the visual cortex and hardly resembles that in the auditory cortex. It's remarkable because all that the cortex knows about the world is contained in the electrical activity of its inputs. There is no independent knowledge that this cortex has as to whether I'm getting visual input or I'm getting auditory input. But from the pattern of activity, from the intervals and the timing, because there's no information in the height of each spike, the brain interprets that as getting a kind of information that should let me make orientation selective cells and orientation maps. So this then leads us to ask, what are these circuits that make orientation selectivity? Hubel and Wiesel proposed a model. There have been a history of models. We have proposed a model as well, along with Chaim Sampolinsky and Shapley. Surya worked with Chaim. And this is a model that says there is a row of thalamic or retinal cells that provides input to a cortical cell, which speak to each other by recurrent connections and amplify that bias. But there must be inhibition to keep the lid on in order to shape tuning and control its gain. And indeed, there is a very important idea in the field of vision, which is that there are invariances. The retina throws away background illumination information and simply retains differences, which is also called contrast. So here's a low contrast grating. Here's a high contrast grating. And similarly, the cortex throws away throws away contrast information and simply retains orientation information. And that's what is shown here. These are the orientation tuning curves of a neuron at different levels of contrast of the grating stimulus. So here the dim grating driving the neuron with different orientations, and this is tuned to a certain orientation, let's say vertical. And then here is a bright grating driving the neuron, and the orientation selectivity is the same, including the width of tuning is the same, so that the range of orientations to which this cell responds is the same. And this is consistent with our behavior, which is that between the outside and the inside, even with these lights on, there must be at least 100-fold difference, maybe 1,000-fold difference in background illumination. It hardly makes a difference to our vision. If I were to wear a dark shirt or a light shirt, hopefully it wouldn't make too much of a difference, even if particularly the background is the same, as to whether you see me better or worse. So illumination and contrast get discounted, and that's what sparse coding is, that the brain codes for differences, not absolute information. And another way to represent this fact is by response contrast curves with orientation as parameter. How might this happen? We think that inhibitory circuits are crucial. Pyramidal neurons are contacted by two inhibitory cell classes. One is called parvalbumin-containing cells. The other is called somatostatin-containing cells. What is their role in orientation processing? This gets us into a little bit of technical detail. But technical development, technology, is of the essence in our field. Just as, so I resonated very strongly with what Sri Kulkarni said. And I don't want to take you through the details, but here's a little glimpse of technology. How can you ask, what does one cell contribute to another? The essential technique in biology is to manipulate and then ask what happened. And so, can we manipulate inhibitory neuron activity and analyze their effect on target cells? How can you manipulate individual neurons? Well, we can express in these neurons or these neurons a gene called channel rhodopsin, and then activate these neurons by shining light on the brain of an animal, a mouse, let's say, in which we have done this genetic manipulation. Molecular genetics is transforming neuroscience. Channel rhodopsin is a molecule that's similar to rhodopsin that exists in our photoreceptors. When light falls on photoreceptors, rhodopsin converts that light into an electrical potential. Channel rhodopsin exists in algae that live in seawater. And that gene has been extracted. It's been packaged into a virus. It can be introduced into certain kinds of mice. And it can get inside parvalbumin cells. And by shining blue light on them, we can activate these cells and only these cells and ask what happened. And together with this, we can use another technique called two-photon imaging by loading cells with indicators that are fluorescent, so that we can use lasers to scan the cortex. Unlike Hubel and Wiesel or my lab earlier used to stick electrodes in the brain and do electrophysiology in order to record spikes. Now we can do electrophysiology with light. So here's what happens. I'll show you a. OK. So we are scanning the brain, the visual cortex of a mouse. There are 72 neurons here. This mouse is looking at oriented gratings. Each blip, I have slowed it down by about 5-fold. Each blip is a spike. You are looking at the electrical activity and the information that is being represented in the brain of this mouse. And in certain trials, here is a neuron that expresses channel rhodopsin. And here's it's a parvalbumin cell, and we excite this cell for half a second on certain trials. And we ask, what does that do to the responses of all these other neurons? And if you were very good at spike counting, you can tell that there are many neurons here that are affected, that the response has gone down due to the additional inhibition. You don't have to count. I'll tell you what happens. Here are visual cortex neurons before and after adding a little bit of inhibition. So this is a tuned response whose response peak goes down, but the tuning does not change. And so what parvalbumin neurons do is that the more the cell's response, the more is the effect of adding a unit inhibition. And that's what this says. And so parvalbumin neurons essentially divide the responses of their target cells. Somatostatin neurons, on the other hand, simply subtract from responses. Their response effect on their target is to suppress equally regardless of the orientation. So they carry out subtractive inhibition. And what this means is that division by parvalbumin neurons alters the response gains. If you were to plot the response contrast function, you've changed the slope of this function. You have changed the peak response, but you haven't changed the tuning width, meaning the half width, let's say, over which the cell responds. Whereas subtraction by somatostatin neurons essentially submerges an iceberg such that the amount that peaks out above the waterline or above some threshold, hence the width of tuning, has altered. So subtraction by somatostatin neurons alters orientation tuning. Hence, in this model, these two neuron types have two different effects. And why is this important? This is a way to implement sparseness, because tuning, as shown by Olshausen and Field, and now gain control, are both, they reduce responses in order to optimize the information coding. So inhibitory neuron circuits create gain control and contribute to tuning. The second question, very quickly, how do cortical circuits create spike codes? Why is this important? It turns out that when neurons are decorrelated, they represent information better. If you have two neurons and they are each putting out 5 spikes, and you are looking at them, and these neurons fire at the same time, there is no information in the second neuron that you can glean. If one neuron fired at slightly different times, if they are decorrelated, you have more information because the second neuron is firing at slightly different times. That's the intuition. It's carried out by acetylcholine circuits in the brain. We know that cholinergic inputs make you more alert, more aroused, and an issue is how. And I'm going to tell you very briefly how this happens. We can take mice in which we can, by molecular genetic means, release acetylcholine in the cortex. We can record the activity of neurons. If you release acetylcholine through this molecular genetic means, you decorrelate the activity. There is more power in the high frequencies, and that is what decorrelation means. And if you record from neurons and you do the pairwise correlations, noise correlations, as it's called, the correlation decreases when you release acetylcholine in the brain. And that's what is shown here, that after acetylcholine, there is less correlation pairwise between neurons. How does this happen? This is a very complicated slide, but here's the way that we think about these experiments and how we do them. You express a molecule called ACh-rhodopsin in just the somatostatin neurons. What is this molecule? When you activate this molecule with green light, it suppresses neurons, rather than channel rhodopsin, which activates neurons. You take a slice, you record from these neurons, and you put a drop of acetylcholine, the neuron fires like crazy because these neurons have a lot of acetylcholine receptors. But then you shine green light on it, and the neuron is completely suppressed, meaning that the ACh-rhodopsin works. You record from these neurons, you put a drop of acetylcholine, these neurons are inhibited. And you shine green light, meaning that you shut down these cells, that inhibition goes away, arguing that a circuit like this must exist. And you record from these neurons and you put a drop of acetylcholine and you see inhibition excitation, suggesting that there is a inhibition-disinhibition phenomena, and you shine green light by shutting down this neuron. You're recording from that neuron and all of that effect goes away, meaning that such a circuit must exist. And why is that circuit interesting? By taking the same animals in vivo, you can shut down the decorrelation and the desynchronization by shining green light and shutting down the somatostatin neurons, even after you release acetylcholine. So you release acetylcholine, the neurons are desynchronized, but you shine green light to shut down the somatostatin neurons, and all of the effect of the acetylcholine goes away. So therefore, blocking somatostatin neurons abolishes acetylcholine-mediated desynchronization and decorrelation, meaning that the system acts through this one neuron type. It's a remarkable amount of specificity. And here's a model that says, why is this interesting? If you have these two neurons, you can decorrelate them by simply removing spikes. But then one neuron is in the limit is not going to fire at all. So that's a boring kind of decorrelation. You want to not reduce activity level, yet decorrelate. And so having a inhibitory, disinhibitory transfer function shifts spike timings without altering the mean spike rate. And we can do this with a very detailed biophysical model to show how it works. So then, inhibitory, disinhibitory circuits decorrelate responses. Do I have 2 minutes? Okay. Finally, how do cortical circuits generate an internal model? And I'll give you the answer because I was concerned that I'll run out of time. Learned visuomotor behavior rapidly engages motor cortex circuits in order to create dynamics that underlie internal representations. We can train a mouse to do interesting things. Here's the simplest task that allows us to probe what is the nature of an internal model. There's a mouse here. My students are like circus trainers. They can train a mouse to do interesting things. They train this mouse to lick when they saw one stimulus and not lick when it saw another. They present a stimulus, then there's a delay where there is nothing in front of the mouse, and then they bring a lick spout forward, and the mouse licks. When it sees this, it doesn't lick when it sees this. Occasionally, it makes mistakes. It sometimes licks when it shouldn't and doesn't lick when it should. I'll show you a brief video, but keep looking here. And there is a mouse here. I hope you can see that. There is a lick spout here. The mouse will see a stimulus. There'll be a delay, and the lick spout will come forward, and the mouse will lick. You can see its tongue going in and out. I hope you see it. OK, so there is the stimulus. There is a delay, 6 seconds, and the mouse licks. OK? So while all this is happening, we are imaging the brain. And we do large-scale imaging. We want to image hundreds, thousands of neurons, because for all the reasons Surya said, we want to record large numbers of neurons. So we express a gene that reads out calcium activity. And then we use our laser system and so forth, and there's a lot of technology that goes into this. We can image from multiple brain regions, and we want to image from the visual cortex, where this image comes in, from the parietal cortex, where we think the delay will be represented, and the motor cortex, where we think the action to lick will be represented. Parietal for the decision and delay, and motor cortex for the action. That's our hypothesis. We can image through many different layers by putting a scanner, by putting a Z-piezo, and we can build systems, and we have built systems for imaging not 1,000 neurons, but 100,000 neurons. We think we can go up to 1 million neurons, and in my students' lifetime, if not mine, we believe we can image every of 50 million neurons in the mouse brain in real time. And we do that by using wide field imaging and deep tissue imaging. And these are all exciting technologies. But very quickly, in one twist in this task, we don't bring the spout forward. So there is a stimulus, is this way or that way, but the mouse realizes there is no spout. So these guys are fooling with me. I should switch off. And during these passive conditions, you can see at a glance that neurons are more correlated. Whereas during engaged conditions, when the mouse is doing this task, it's highly attentive, there is decorrelation. So engagement decorrelates responses. And here is a little bit of data. And the astonishing thing is that here are the visual cortex responses, which you see when there is the visual stimulus and then nothing else. So these are visual neurons. The parietal neurons respond to the visual stimulus or sometimes to the lick. but not in the delay, to our astonishment. It is the motor cortex that carries the full information for this task. Motor cortex activity goes up. Even when the visual stimulus is on, the motor cortex begins to fire. And then it holds that activity during the delay. Remember, during the delay, there is nothing in front of the animal. There is just the imagination of the mouse. I must lick as soon as these guys let me lick. But the brain knows it, and the brain carries that information. And then it licks when it can. And of course, we can use dynamic systems analysis and PCA for these hundreds or thousands of neurons to see where is the information. And it is the motor cortex that carries the internal state and action. And that's what it looks like. That here are the trajectories. Here is and here is the basin of the memory. So when the stimulus comes on, there is a set of neurons that fire. They hold the action in memory, and then a set of neurons licks. And 80% of the variance in this very simple task can be captured by these kinds of ideas. And so this predicts a model like that. It's a well-known model of how memory is created. What is being held in memory? I don't have time to tell you, but there is strong evidence that it's actually a motor plan. Because as soon as the stimulus comes on, the visual cortex shoots information forward. That's what learning is. So there are deep questions in our field. What does it mean to learn? What does it mean to see? What does it mean to have a reinforced learned behavior? And one thing that it means is information shoots forward to where it can be used. And it can be used in the motor cortex, where a motor plan is created right away during the stimulus and is held in memory and then used to execute. What have we learned so far from all this? And I'm almost finished. The brain interacts with the world at different time scales. Of course, through evolutionary time scale. We are products of evolution. Through developmental time scale. And I showed you how we can rewire brains, and the world can write on the brain, but it doesn't write on a blank slate. The rewired auditory cortex is not identical to a visual cortex. There is always a slate. The idea of the brain being a mirror of the world is absolutely not true. But the brain does interact with the world. The world shapes the brain, and the brain in turn shapes the world. It's a deep dialectics in terms of understanding brain function. And of course, we live in the present, and the present is represented in our brain through memories and through working memories. And of course, the brain reflects on the future or makes predictions about the future. In fact, half of perception has to do with interpolation. We think we see everything that we react to. In fact, we don't. We just interpolate all the time. The brain processes only a fraction of the information it receives. I gave you very simple examples of the retina throwing away illumination, of the cortex throwing away contrast. Inhibitory circuits regulate this deep idea of sparseness. They reduce and reorganize by decorrelation, and hence perhaps maximize, though this remains to be proven. And this is yet another way where we think theory has a great deal to offer. The brain does not simply mirror the world as activity and dynamics create an internal model of the world. Our work, the example Surya gave, they're all about internal models. How do we represent a task? How do we represent components of the world? The dynamics of large numbers of neurons. And cognition reflects these internal models. And finally then, the brain creates cognition, and so here are some issues. We can state the brain creates cognition by dynamically extracting, representing, and transforming information. Our goal is to understand the human brain. We can hardly study the human brain. We can only do non-invasive measurements, or in a few people who give you access because they are desperate to the open brain, so to speak, so that you can put electrodes. But that's very few. So we use model systems. Here is a set of connections of the monkey brain showing how many visual areas there are. The mouse brain is 1,000th the volume of a human brain. It's nothing like a human brain. It has some regions that are homologous, but the mouse brain is not a miniature version of the human brain. A mouse will not write Shakespeare. So we study what we can. And a great challenge for our field is how can we derive inferences from the limited information when the critical kind of information we need needs needs access. Many theoretical questions from the top down. What is cognition? What is the nature of this complex system? What is causality in the brain? The useful levels of questions to my mind are at the mesoscale. What are the nature of representations? What do dynamics convey? What does it mean to transform? Even what is information for biological systems? Before you can decide what is information, you must have information about what does it do? Why does it exist? And that is a little different, as Sanjay and I have talked about, from pure Shannon information. And then at the deepest level, there are questions of biophysics of neurons and how individual neurons work, how their properties via synapses and the ion channels and the proteins get made and so forth. A deep theory would integrate these levels one day. And of course, it is wonderful to ask questions about our place in the universe and the nature of matter, but it's our brain that lets us do so. So understanding our brains may give us some insights into many other things as well. Thank you very much.
Okay, we are running a little bit late, but
so these are the optogenetic experiments that you have been carrying out, right?
The experiments that I described are optogenetic experiments, yes.
So have you been trying these things which people have been doing about creation of memories, creation of false memories?
Oh, yes, of course. With optogenetics, you can induce percepts. And that tells you that certain neuronal groups are important. What you cannot do yet is that optogenetics is an all or none kind of experiment, or you cannot create the activity pattern necessarily of the real brain.
Yeah.
And once we are able to do that, that would be even more promising, in my opinion. Yeah. So optogenetics is a kind of a crude experiment, but it's impressive how well it works when it works. And when we are able to mimic the actual dynamics of the brain by stimulating neurons with light, but artificially, those will be powerful existence proofs that these neuronal codes matter. A lot of neurophysiology is really about correlations. It's not about causality. When we record from neurons, we are correlating the activity of these neurons with the animal's behavior. Optogenetics tells us a little bit about causality, but there are still open questions because the brain is not just one region, it is an interconnected network. And so in what way does the network matter? To what extent does pure activity independent of timing matter? These are questions that optogenetics will need to address, if ever, and may not be able to address ever.
See, my other question has to do with correlations. You said that the lack of correlation is actually better, but you are talking of lack of correlation in the input, right?
No, I'm talking about lack of correlations in the representational space, where you have independently active neurons provide more information in a sense. When you have correlated neurons, then there is not independent information. That's a simple-minded view of why decorrelation is good for information representation. But correlation is good for information transmission. If you have two neurons that are connected to one neuron, if they fire together, then the probability of the next neuron firing is higher than if they don't. But if you are looking at what is the information in this population, if two neurons are all firing at the same time, they are highly correlated. There is not independent information in them.
If you look at the same neuron and you look at temporal correlations in the series.
These are temporal correlations.
But at different times, the same neuron.
You mean over time slices? They do change.
They do change.
Yeah, they do, of course. Yeah.
And they do carry information.
Yes, they do.
okay, if not Any questions? Thank the speaker again.